Okay, in this video, we are going to look at sex-linked inheritance. First, let's look at the meaning of sex-linked genes. Okay, this refers to the genes that are located on the sex chromosomes, which control specific characteristics but are not involved in sex determination. Genes of color blindness and hemophilia are located in the X chromosomes. And these genes are called the sex-linked genes. Characteristics of color blindness and hemophilia is caused by, it is important here, it is caused by the recessive genes that are linked to the X chromosome. Y chromosome is shorter than X chromosome. Okay, let's look at this figure again. This is X chromosome and compare that this is the Y chromosome, which is much shorter, okay, and does not contain as many alleles as the X chromosomes. Therefore, any traits in males caused by either the dominant or the recessive allele on the X chromosome is observed. Okay, so let's look at the first condition, and that is color blindness. Okay, this condition happens in which a person, this is a condition in which a person cannot differentiate some specific colors, such as red and green, merah dan hijau. Color blindness is caused by recessive allele, which is found in the X chromosome. And most people with color blindness are males. Okay, so in the sex-linked inheritance research, X and Y chromosomes must be shown when writing the genotypes. The dominant allele is represented by a capital letter huruf besar, whereas recessive alleles is represented by a small letter huruf kecil on the X chromosomes. Now let's look at the genotypes of the color blindness as shown here in the table. Okay, first, let's again look at the key indicator. If it is X and then capital B, this represents the dominant allele. But if it is a small b, this represents recessive allele. Okay, for a male with a normal eyesight, so that means this XB, this is capital B, meaning the absence of recessive allele, therefore the male is having normal eyesight. Okay, same goes to the female with homozygous dominant allele. Okay, carrier, only female can become a carrier in which the female is having heterozygous for color blindness, so she is a carrier. Okay, colorblind, a male will be colorblind whenever he is carrying a recessive allele at the X chromosomes. But for a female to be colorblind, it must be homozygous recessive for the X chromosomes. Okay, so this is a picture showing the schematic diagram when, let's look at the father. A normal eyesight, a man with normal eyesight marries a woman who is heterozygous for color blindness. Okay, so first step of doing the schematic diagram is we write down the phenotype. Normal eyesight father, normal eyesight mother, but then the father, the father is normal, but the mother, despite having normal eyesight, she is actually a carrier because of the genotype. Let's look at the genotype. There's a small letter here. There's presence of a recessive allele, but then it doesn't show the effect. So she is a carrier. Okay, after writing down the phenotype, genotypes, and next is the process of meiosis. Okay, so you write the, as usual, the arrow, and therefore formation of gamete, where only one allele is being inherited from the pair. So as we break it down, this is what we get. Fertilization takes place where this X capital B from the father fertilized, let's say these are the probabilities, okay, fertilized with the the capital B from the mother. So therefore the outcome is this, the child genotype being homozygous dominant. Okay, so X capital B to fertilize with the recessive allele from the mother. So this will be the child genotype. This is heterozygous for color blindness, which means the daughter XX is a girl, XX is a girl. So that means this daughter is a carrier. Okay, if Y, this Y gamete from the father is to fertilize with the capital letter, okay, that means dominant allele from the mother. So the child will be a boy with normal eyesight. But let's look at the last probability where the Y from the father fertilized with the recessive allele from the mother. So X, small letter, B, Y. So this is a boy with color blindness. And therefore, okay, this is number 6. The genotype of the child, the phenotype of the child. Last but not least, we are to show the phenotypic ratio. Okay. Now this picture, this figure 11.20 is an example of Ishihara test plate by the name Ishihara. Okay, sounds Japanese. Okay, Ishihara test is a famous color blindness screening test that has been used worldwide since 1917, where the test was developed by Shinobu Ishihara. He was a Japanese ophthalmologist, and it is invented to screen for the common green-red color blindness defects. Okay, if the individuals with they have normal eyesight, okay, the individuals can easily identify the numbers. Okay, so can you identify the numbers that is shown here? Okay, they can easily identify the numbers or the pattern in the Ishihara test plates. Whereas individuals with color blindness defects would report wrong numbers or fail to identify the numbers and patterns.